Hello everyone, it's Mimi here. BMS working in the NHS. And today's video will be about molecular analysis of sample homogenates, which is part of the IBMS specialist portfolio in cellular pathology. So, just a quick introduction to this video. It will be short and sweet, I promise. I will discuss indicative repertoire, knowledge requirements, competence requirements, reflective statement, evidence of achievement, Q&A suggestions, evidence ideas, and some helpful tips to make candidates' portfolio really good. With all that said, I hope you will enjoy the content of my video and I hope you will learn a lot. So let's dive into the second subsection of the fourth section, molecular testing, called molecular analysis of sample homogenates. The portfolio offers indicative repertoire for this section that includes BRAF V600E, CRAS, EGFR, ROS1, T-cell gene rearrangement studies. The knowledge requirement explains what knowledge is expected of the candidate in relation to the relevant subsection. Candidates should be able to demonstrate their knowledge at the specialist level. Think about what knowledge they had as a Band 5 BMS. How would that differ to the knowledge of a Band 6 BMS? That specialist level of knowledge would allow the candidate to troubleshoot, problem solve, verify or validate. The first knowledge requirement is principles of molecular biology methodology. Candidate needs to have the knowledge and understanding about molecular testing. You could ask the candidate to explain what molecular testing is, to name molecular techniques that can be used to assess histological tissue, to explain how those methods work, to name sample types that can be used for molecular analysis. When addressing this knowledge requirement, candidates should consider molecular methods like PCR, sequencing, microarrays, blotting, mass spectrometry, their principles, tissue preparation for those methods, detection of genetic mutations, nucleic acids, and proteins, advanced imaging, methods stated in indicative repertoire and methods used in your lab. The second knowledge requirement is clinical value and application of molecular biology techniques in diagnostic cellular pathology. Candidate needs to have the knowledge and understanding about value and application of molecular testing. You could ask the candidate to explain the clinical value of molecular techniques used in histology, to list their applications, to explain how they are used for diagnosis, prognosis, monitoring, or treatment decisions. When addressing this knowledge requirement, candidates should consider diagnostic accuracy, use for diagnosis, prognosis, disease identification and monitoring, guiding therapy, role in personalized medicine, cancer and infectious diseases, monitoring treatment, detecting hereditary diseases, identify predisposition and guide preventative strategies, types of molecular technique and their value, advantages and disadvantages. The third knowledge requirement is role of molecular methods using sample homogenates in the diagnosis, prognosis, therapeutic implications of key named pathologies. Candidate needs to have the knowledge and understanding about role of molecular methods in diagnosis, prognosis, and treatment of diseases. You could ask the candidate to explain the role of molecular methods using sample homogenate in diagnosis, prognosis, and treatment of key pathologies, to list key pathologies that can be assessed using those methods, to give examples of methods that use sample homogenate. When addressing this knowledge requirement, candidates should consider various molecular methods that use sample homogenate, the role in classification and subtyping diseases, detection of genetic alterations, differential diagnosis of ambiguous tumors, detecting unknown primaries, the role in prognosis like estimating progression and aggressiveness of disease, detecting micrometastasis, the role in treatment and guiding personalized or targeted therapy, minimizing toxicity from drugs. Competence requirement is basically a combination of knowledge, skills, and experience candidate needs to perform a job to a satisfactory standard. Candidates' chosen evidence should demonstrate these competence requirements. The following competence requirements need to be met for this subsection. The first competence requirement is prepare sample homogenates from cell samples for DNA and RNA analysis. Candidates should be able to prepare sample homogenates for DNA and RNA analysis. Does the candidate prepare sample homogenates for DNA and RNA analysis? Do they take correct steps and why they take them? Do they follow local protocols and SOPs? Do they correctly troubleshoot problems encountered during sample preparation and DNA and RNA analysis? When addressing this competence requirement, candidate could consider sample size, handling, temperature control to preserve genetic material, mechanical, genic or sonical homogenization of sample, lysis, extraction of genetic material, separation, different methods for fresh tissues, use of sterile plasticware, cleaning with bleach, ethanol, minimizing cross-contamination, sample stabilization for delayed processing. The second competence requirement is select and use control material, maintaining appropriate audit trail. Candidate should be able to select and use correct control material. Does the candidate use correct control material? Do they maintain correct audit trail for control material used? When addressing this competence requirement, the candidate could consider tissue type and availability, verification and validation of control material, selecting correct control material, commercial kits available and their control material, record keeping, traceability, audit trail, consequences of not using correct controls. The third competence requirement is assess preparations for nucleic acid integrity and purity. Candidate should be able to assess preparation for nucleic acid integrity and purity. Does the candidate assess nucleic acid integrity and purity of preparations? Do they use various methods for the assessment? Do they follow relevant steps during the assessment? When addressing this competence requirement, candidate could consider spectrophotometry for assessing purity and detecting contamination with proteins, salt and solvents, use of fluorometry for purity via comparative quantification, use of electrophoresis to assess integrity of nucleic acid either via RNA integrity number in fresh tissue or via DV200 metric for RNA fragments longer than 200 nucleotides, quantitative PCR to assess functional integrity, spectrophotometry and fluorometry for purity, electrophoresis and quantitative PCR for integrity. The fourth competence requirement is undertake qPCR analysis for identification of specific gene sequences. Candidate should be able to perform PCR analysis. Does the candidate undertake PCR analysis for identification of specific gene sequences? Do they use commercial kits? Do they relevant steps? Do they understand the method they perform? Can they give examples of gene sequences they can identify? When addressing this competence requirement, candidate could consider their training and competence, SOPs, methodology used, sample preparation, denaturation, annealing, extension, specific gene sequence, commercial kits available to detect KRAS, BRAF, ROS1 and others. The fifth competence requirement is resolve problems associated with the demonstration methods, including root cause analysis, as appropriate. Candidate should be able to troubleshoot demonstration methods. Does the candidate resolve problems with demonstration correctly? Can they identify and solve the common problems? Can they record problems and resolutions accurately? Can they perform root cause analysis? When addressing this competence requirement, candidate could consider investigation, problem identification, corrective actions to fix the issue, preventive actions to minimize problems from reoccurring, possible problems with sample degradation like thermal, endotic or oxidative, incomplete lysis, low yield, contamination, interference, technical and methodological limitations, sample-specific challenges like fatty or tough tissues, blood contamination, fragmentation of genetic material. The sixth competence requirement is dispose of waste reagents and residual material safely. Candidate should be able to safely dispose waste generated. Does the candidate dispose detection reagents and residual material safely? Do they follow local procedures and avoid risks associated with detection reagents? Segregate waste as per regulations? When addressing this competence requirement, candidate could consider licensed contractors used by your lab, waste segregation, safety data sheet of reagents that often state disposal ways, risk assessments, local policies and regulations that are followed, health and safety risks associated with reagents like stability, corrosivity, environmental toxicity, risks associated with disposal or residual material like human tissue. Each section also contains reflective statement which is asking the candidate, to summarize their role in the lab and the context with each section. When they complete all the subsections within each section, they will be required to write a reflective statement. They also need to write a personal reflection on their training and why they chose their evidence. To prepare for that, ask them to write a smaller reflective statement for each subsection. What is their role and context of this subsection within the lab? What does their evidence demonstrate? Ask them to write a smaller justification as to why they chose their evidence for this subsection. Also ask them to write about what was their training like when to this subsection. Writing a smaller reflective statement at the end of each subsection will then make it much easier for them at the end of the section. Each subsection also has evidence of achievement. This is a part where you, the training officer, sign the candidate off as competent and fit to practice as a BMS at the specialist level. By doing that, you will confirm that the candidate can work in accordance with local SOPs relevant to this subsection. This will also confirm that candidate has answered questions set by you to test their knowledge and skills relevant to this subsection. And you will also confirm that candidate's chosen evidence demonstrates their competence relevant to this subsection. The candidate will need to be signed off as competent by an internal assessor for each subsection. They can be signed off by you, the training officer, senior BMS overseeing relevant section, or by the training lead. These are some example questions you could give your candidate to test their knowledge and skills about this subsection. These should be based around the knowledge and skill requirements mentioned earlier. These are some more example questions you can ask your candidate to complete, to test their knowledge and skills about this subsection. You could ask the candidate to complete tables you created, based on knowledge requirements. Supporting evidence should be selected by the candidate. It needs to be based around the competency requirements, and be relevant for each subsection. The evidence should allow the candidate to demonstrate their competence in each subsection. It should be specific and relevant to the candidate's in-house practice. It should also demonstrate candidate's specialist competence. If you would like to see specific ideas for supportive evidence, please have a look at the video for trainees. Link will be included in the video description. I would also like to share with you some helpful tips that can make candidates' portfolio look really good. Ask them to try and present the information in various formats such as tables, pictures, photographs, diagrams, flowcharts, graphs, or even videos, if they can. This nicely breaks down a lot of text that is usually present in specialist portfolios. Ask them to try and make it a little bit more interesting to the reader, which will be you, and eventually the assessor. Ask them to try and put themselves in your or the assessor's shoes. How would they like to read hundreds of pages of plain text? There is a saying I really like, and it goes like this: a picture 1,000 words. I will make a video where I will share with you my evidence I used for my portfolio back in the day. Hopefully that will help. Also ask them to try and use various types of evidence, not just one type, like case studies, for each subsection. They could use experiments, case studies, annotated results, annotated photographs of them performing tasks, evidence of troubleshooting, annotated EQA results, nonconformances, validation verification evidence, comparisons of different approaches and their pros and cons, control material production, certificates of course attendance with reflective statements, annotated images or charts, and so on. Ask them to try and bring in their personality. Ask them to think about the ways they like to learn best. If experimenting, let them perform experiments. If they like troubleshooting, let them perform troubleshooting. Ask them to choose something that demonstrates their specialist understanding, knowledge, and skills, and how they apply this in their practice. And one more thing, ask them to keep the competence requirements in mind when they are selecting their evidence. Good evidence will tick off several requirements. And this brings me to the end of today's video. Hopefully you now have a better understanding about the knowledge and competence requirements for this subsection. And I hope you also have a better idea what questions you can choose to test candidates' knowledge. And my final take-home message? Keep those requirements in mind when selecting questions and guiding the candidate. Bye for now.